Hey there and welcome. In this explainer, we are going to absolutely nail the microscopy required practical, breaking it all down step by step so you feel totally ready for your AQA GCSE Biology exam. Okay, let's kick things off with a bit of a weird question. What on earth could you possibly have in common with an onion? And no, it's not a trick. Just think about what makes up all living things. You got it. You're both made of cells, these tiny, amazing building blocks of life. And to actually see this hidden world, we need a special tool, the microscope. It's pretty much our window into a whole new universe. So here's our game plan. First, we'll look at why we even use microscopes. Then we'll get practical, covering how to prepare slides and get a perfect focus. After that, we'll dive into scientific drawings and magnification maths, and we'll wrap it all up with some top exam tips. Right then, Section 1, why do we bother with microscopes? Well, it all boils down to two really important ideas you've got to know for your exam: magnification and resolution. First up, magnification. This one's pretty straightforward. It's just how many times bigger the image you're seeing is compared to the actual object. You can think of it like the zoom on your phone's camera. But, a huge image is totally useless if it's just a giant blur. And that's where resolution comes in. Resolution is all about the clarity, the ability to tell that two separate points are, well, separate. A high resolution gives you a super sharp, detailed image. This is really the main difference between our school light microscopes and the mega powerful electron microscopes that can see tiny things in crazy detail. So what are we actually looking at? Animal and plant cells. As you can see, they both have a nucleus, cytoplasm, and a cell membrane, but you've got to know the differences. Plant cells have that extra rigid cell wall for support, a big permanent vacuole, and often chloroplasts for photosynthesis. Okay, let's get stuck into Section 2. This is the hands-on part, the practical method. This is literally what you'll be doing in the lab, so pay close attention. So to make your onion slide, you start with a single drop of water on a clean slide. Then you'll need to peel off a really thin, almost transparent layer from the inside of the onion. That's the epidermis. Place it flat in the water. Next, add a drop of iodine. The stain is brilliant because it makes the nucleus and other parts of the cell actually show up. And finally, and this is super important, gently lower the cover slip on top at an angle. Doing it this way helps to prevent any annoying air bubbles from getting trapped. Now, to get that image in focus, first, clip your slide securely onto the stage. Now, always start with the lowest power objective lens. That's the shortest one. Use the big coarse focus knob to move the stage all the way up. Then, looking through the eyepiece, you slowly turn the coarse knob to move the stage down until you see something. Once it's roughly in focus, you just use the little fine focus knob to make the image perfectly sharp. And here is the golden rule. When you switch to a higher power lens, you only use the fine focus knob to adjust it. Got it? All right, Section 3. You've found your cells, you've got them in focus, now what? You need to record what you see. And in biology, that means doing a proper scientific drawing. This is such a key skill, and it's worth a lot of marks, so let's make sure you know how to do it perfectly. The rules are non-negotiable, but they're easy to remember. Always use a sharp pencil. Draw with clear, single lines, no sketchy, hairy lines, and absolutely no shading. Make your drawing nice and big so you can actually see the details. Keep everything in proportion. Use a ruler for your label lines to keep them neat. And finally, always include a title and the magnification you were using. Follow those, and you're golden. On to Section 4, and it's time for a little bit of maths. But please don't panic. This is genuinely one of the easiest ways to bag yourself some marks in the exam, as long as you learn one simple formula. And here it is. Image size equals actual size times magnification. So the image size is the length you measure on your drawing. The actual size is how big the cell is in real life, and the magnification is just how much bigger you've made it look. And to make rearranging that formula an absolute breeze, use this formula triangle. A good way to remember it is IM. Just cover up the letter you want to find, and the triangle tells you exactly what to do. So if you want to find magnification, cover up the M, and you're left with image size divided by actual size. Easy. Okay, let's put it into practice with a classic exam question. An image of a cell is 50 millimeters wide, and its actual size is 0.05 millimeters. So what's the magnification? Right, step 1, we use our triangle to get the right formula. Magnification equals image size divided by actual size. Step 2, we just pop the numbers in. That's 50 divided by 0.05. Now here's a really crucial check. Make sure the units are the same.
Here, they're both in millimeters, so we're good to go. Pop that into your calculator, and the answer is a magnification of times 1,000. See? Not so bad when you break it down.
And that brings us to our final section: how to stop throwing away easy marks. Let's run through the most common mistakes students make so that you can avoid them. You really want to watch out for these traps. Air bubbles are a classic. They just get in the way of what you're trying to see. Never, and I mean never, use the coarse focus knob on high power. You could smash the slide right into the lens. It's so easy to forget a title or magnification on your drawing, but that's a lost mark right there. And of course, double-check those units in your calculations. Avoiding these simple slip-ups makes all the difference.
And here is an absolute gem of advice straight from the examiners. Always show you're working out. Seriously, even if you have a calculator malfunction and the final number is wrong, you can still get marks for showing the right method. It's a safety net, so make sure you use it.
So to wrap it all up, what are the key skills? You need to prepare a clean, bubble-free slide. You need to focus carefully, going from low to high power. You need to draw a big, clear diagram, following all the rules. And you need to confidently use that I am triangle to smash your calculations. And that's pretty much it. You're now armed with the skills you need to go and explore the microscopic world. It kind of makes you wonder, doesn't it? Now that you can see cells, what other incredible invisible worlds are just out there, waiting to be discovered?